Hi, my name's Skylar and today we'll be talking about prokaryotic genetic regulation. Here we see a diagram of the operon. The operon consists of the key features necessary for transcription. The promoter is the landing pad for RNA polymerase, and the operator is the landing pad for a regulatory protein. There could be multiple genes that are encoded for within an operon, as you see here. Structural genes A, B, and C here are transcribed and translated into proteins used in metabolism. They can also play a structural role in the cell. Similarly, the regulatory protein, whether it's an activator or repressor, is encoded for by the respective genes. In this case, we'll be looking at positive repressible regulation specifically, where an activator protein is present. One example of a regulation is positive repressible control. In positive repressible control, if the effector is present, it binds to the activator protein, making it impossible for transcription to occur. If there is no small molecule to bind to the activator binds to the activator binding site, which causes RNA polymerase to come in and carry out transcription as normal. Here is an analogy depicting positive repressible regulation. Some individuals have peanut allergies where normally, if they eat peanuts, they go into anaphylactic shock. However, if they inject themselves with epinephrine, they can prevent this anaphylactic shock from occurring. In this case, anaphylactic shock represents transcription, the peanuts represent the activator, and epinephrine represents the small molecule or the effector. Since the regulatory protein is an activator, this is positive control. This also means that transcription occurs when the protein is bound to DNA, or in this case, when a peanut is ingested, anaphylactic shock occurs. We also know that this situation is repressible because transcription is turned off when the effector is present, or in this case, anaphylactic shock stops when epinephrine is present.